is a double helix made up of nucleotides. Nucleotides are composed of a phosphate, a deoxyribose sugar, and a nitrogenous base. The sugar and phosphate are found in a chain on the outside of the molecule and make up the backbone of DNA. The nitrogenous bases like adenine, guanine, cytosine, and thymine stick out in the center of the molecule like rungs on a ladder. Remember that adenine and thymine pair together, and cytosine and guanine pair together. The specific order of the nitrogenous bases is the genetic code that determines an organism's traits. It is very important to maintain the specific order of bases that make up the genetic code of an organism. If the genetic code is altered, the organism may not possess the genetic traits it was intended to have. Most of the time, the genetic code of an organism is fixed and cannot change during its lifetime. Occasionally, however, certain environmental factors can cause the genetic code to change. Any sudden change in the nitrogenous base sequence of DNA is called a mutation. How does a mutation affect an organism? Recall that most of our physical traits are coded for in the genes located on our DNA. A gene is like a blueprint that codes for a sequence of amino acids that forms a protein. If the DNA sequence changes as it does in a mutation, the amino acid sequence could be different too, and the resulting protein might not function properly. Let's take a look at a few different types of mutations and how they affect the proteins coded for by the gene. A point mutation involves a change in a single nitrogenous base in the DNA sequence. When one base is switched for another, it is called a substitution. Let's take a look at how a substitution in a gene might affect the protein it codes for. In this case, the third base in codon 3 has been changed from a G to an A. The base substitution changed the codon, but the new codon still coded for the same amino acid, so the protein will be unaffected by this particular mutation. When the protein is not affected by the mutation, it is called a silent mutation. Here's another example of a substitution. This time the base was changed from G to U. The new codon codes for asparagine instead of glutamate. This could potentially cause a change in the protein that could change its function. In this case, the point mutation occurred in codon 2, changing the C to A. The codon UAG is a stop codon. This mutation caused the peptide chain to end prematurely. This could dramatically affect the function of the protein, or, depending on where the premature stop occurs, might not have much of an impact at all. Another type of mutation is called an insertion. An insertion mutation occurs when an additional nucleotide is added to the messenger RNA strand. A deletion mutation is just the opposite. During a deletion mutation, a nucleotide is deleted from the messenger RNA strand. Both insertion and deletions are examples of a frame shift mutation. Frame shift mutations shift all of the nucleotides of the messenger RNA strand. Remember that messenger RNA is read in sets of 3 nucleotides called codons. A frame shift mutation can change the way the codons are read, and in turn, change the sequence of amino acids in the protein. In this example, you can see the effects of a frame shift mutation. The deletion of the base uracil caused all of the other nucleotides to shift to the left. All the codons following the deletion have been changed, so that in turn changed the amino acid. You can see that after a deletion mutation, an entirely different protein is manufactured. The same is true of an insertion. Not having a functional protein could potentially have a great impact on an organism. Albinism, for example, is the result of a mutation that results in a defective enzyme responsible for producing melanin, the pigment in skin. Because the enzyme is defective, no melanin can be produced, which leads to the lack of pigment that characterizes albinism. Albinism can lead to vision problems and an increased risk for skin cancer, but it is not usually a deadly mutation. Other mutations, however, can be lethal to an individual. Yet another form of a mutation occurs when the bonds of the sugar phosphate backbone of the DNA molecule is broken. If the break occurs within a gene, then that gene will no longer function properly and the cell will no longer be able to manufacture the protein coded for by the gene. Cells with broken DNA try to fix the broken section by joining it to other pieces of DNA within the nucleus. Sometimes the broken piece can get fused to the wrong chromosome. When non-homologous chromosomes exchange genetic material, it is known as a translocation. Translocations may cause a gene to function abnormally and therefore have an impact on the organism. Now you've been introduced to some of the different types of mutations. But what is it that causes these mutations to occur in the first place? Many things can cause a mutation to the DNA in your cells. Environmental factors such as radiation and certain chemicals or pollutants can cause mutations. Any agent that increases the rate of mutations in DNA is known as a mutagen. Specific examples of mutagens include UV light, X-rays, certain chemicals in cigarette smoke, and carbon monoxide from car exhaust. Mutagens that specifically lead to cancer are called carcinogens. UV light, for example, is a carcinogen that causes skin cancer. Other examples of carcinogens are pesticides, asbestos, and formaldehyde. Mutations that occur in gametes, like egg or sperm, can be passed on by the affected parent to their offspring. In this way, mutations can become hereditary. Mutations that occur in body cells other than gametes are called somatic mutations. While these mutations can be harmful, they only affect the individual where the mutation has occurred. Somatic mutations will not be passed on to offspring, and so their impacts are not quite as far-reaching as mutations to gametes. Some examples of mutations that have become hereditary conditions are hemophilia and sickle cell anemia. Hemophilia is a blood disorder in which the blood fails to clot after an injury. Just a small wound could allow a person with hemophilia to bleed to death. The mutation that causes hemophilia is found on the X chromosome. It is also, luckily, a recessive disorder. So hemophilia is an example of a sex-linked recessive disorder. Recall that females have 2 X chromosomes, where males possess 1 X and 1 Y. Sex-linked recessive disorders are more common in males since they do not possess a second X chromosome that can counteract the effects of a mutated one. In order for a female to inherit hemophilia, she would have to have 2 recessive alleles for the disorder. This makes sex-linked recessive disorders like hemophilia more common in males. Sickle cell anemia is caused by a mutation to the gene coding for hemoglobin, a protein in blood that carries oxygen. In people affected by sickle cell anemia, the blood cells take on a sickle shape and have a greatly reduced ability to carry oxygen. Sickle cell anemia is a recessive disorder. For people who are homozygous recessive for sickle cell, the condition is usually deadly. So how do genetic diseases like hemophilia and sickle cell anemia stick around? You would think that potentially fatal diseases would get weeded out of the gene pool by natural selection as the affected individuals died. Both sickle cell and hemophilia are recessive disorders that can be hidden by a dominant gene. Individuals who are heterozygous for such conditions are known as carriers, and though they do not display symptoms of the disease, can still pass it on to their offspring. Additionally, with certain genetic disorders like sickle cell anemia, it can actually be beneficial to be heterozygous for the disorder. This is known as heterozygote advantage and is a huge reason why sickle cell anemia persists in the population. You see, individuals who are heterozygous for sickle cell anemia are less susceptible to malaria, a disease caused by a blood parasite. Malaria is common in tropical regions, such as those found in Africa. This is why sickle cell anemia occurs almost exclusively in individuals of African descent. Being heterozygous for the sickle cell condition actually gives them an advantage for survival. How interesting! As you can see, genetics is a really intriguing field, particularly when it comes to mutations. The next two lessons are labs that focus on some of the techniques that geneticists use to conduct their work. Perhaps this is a field you might consider for a career.